Hello, everyone. Welcome. Um, Everybody's coming in. Looks great. So hi, welcome to the Moss Landing Marine Laboratories seminar series, now online. So I am Amanda Kahn, and on behalf of the Invertebrate Ecology Lab, today we'd like to welcome you all and our speaker. So really quickly, how this online seminar will work. All audience participants will be muted throughout the seminar. Please do not attempt to turn on your video or share your screen during the talk. There will be an opportunity for the audience to ask questions live at the end of the talk, just like in a normal seminar. So once Dr. deVries has finished her talk, you can use Zoom's raise hand feature to notify the meeting host that you'd like to ask the speaker a question. So the raise hand feature is located under the participants tab at the top of the Zoom window. You can take a moment to find that for later if you like. Also, please keep your time after the seminar open as well. Dr. deVries is available to stick around for a more informal happy hour and discussion following the seminar and the question period. So with those logistics, I'd like to introduce Dr. Maya deVries. She got her bachelor's degree in evolution and ecology at UC Davis. And then she pursued her PhD in integrative biology at UC Berkeley under co-advisors Dr. Sheila Patek and Todd Dawson. And there her thesis was on the ecology and morphology of stomatopod crustaceans. Since that time, she's been a visiting fellow at the Smithsonian Tropical Research Institute in Panama, a postdoctoral fellow at Scripps Institute of Oceanography, and the director of training activities at the Coastal and Marine Studies Institute at UC Davis, before starting up in the biology department at San Jose State University at the beginning of this year. So Dr. deVries' research has covered a broad range of animals and questions, with a general focus on the ecological and the evolutionary drivers of different modes of feeding. She's used functional morphology and a diversity of approaches to understand mechanisms of feeding in animals ranging from mantis shrimp to largemouth bass to sea urchins. Her lab has 5 student members already, all studying how feeding can shape the trophic dynamics of an ecosystem. Her research has often appeared in the popular press because she is a committed proponent of outreach, education, and science communication, and because, well, her research is just pretty darn cool. So with that, I am looking forward to hearing Dr. deVries present her research today. Her talk is entitled Trophic Relationships in the Benthos, Feeding Morphology and Ecology of Macroinvertebrates. Dr. deVries, welcome.
Thank you so much, Dr. Kahn. That was such a lovely introduction. I am so excited for the opportunity to share my research with you all, and it's really an honor to get to speak to the Moss Landing community. I was told that generally for these talks, the speaker keeps their cameras off because it looks a little bit better in the recording, so I will do the same, but I will turn it on at the end for questions. So I'd like to start by showing you this photo of a prey capture event in stomatopods or mantis shrimp. And so here we see this mantis shrimp lunging out of its sandy burrow with these spear-like appendages to capture this damselfish. Now, this spear-like appendage has certain modifications or specializations, like multiple spikes on the appendage to help it spear its prey. And this animal is in the process of doing something that all animals have to do, and that is consuming food resources. So this trophic interaction demonstrates the fundamental need to feed, and that is an important factor for shaping the amazing global biodiversity seen in the world. It is for these reasons that my research is broadly interested in the relationship between morphology and ecology and how that relates to trophic dynamics. And so the reason why I'm telling you about mantis shrimp is because they're really an excellent example of an animal with specialized morphology, and one that we thought we knew a lot about in terms of its ecology, but as I'll tell you, these animals are actually quite surprising in their trophic ecology, and they influence their environment in ways that we hadn't expected. And so you see they're quite speciose, about 500 extant species. We find them in the tropics and the subtropics, and they have these appendages that move extremely fast. and specialized feeding ecology, which we'll talk about today. And so many stomatopods or mantis shrimp live in these coral reef habitats. And you wouldn't know it from looking at this picture, but this reef has millions of species in it. Occupying less than 1% of the seafloor, coral reefs are home to more than 25% of all marine life. And these organisms are interacting with one another and their environment, and they're consuming food resources. And additionally, they're also an environment that's rapidly changing. And so the reefs themselves are undergoing unprecedented changes, and many of them are dying and unable to recover. So given the importance of the many consumers on reefs and the habitats and habitats in general, my perspective is really that we can't understand ecosystem function holistically without a deeper knowledge of morphology on the individual level, feeding interactions between organisms, and how that relates to the trophic dynamics and environment on the ecosystem level. And so today we're going to explore this specifically in mantis shrimp, but then broaden this to a reef-wide scale and also examine these ideas in a few other organisms that are more local and ones that you are probably all familiar with as well. And so on a fundamental level, I am interested in how animals capture food, how these interactions fit into trophic dynamics, and how these feeding strategies may shift under future ocean conditions. And so these three topics, feeding interaction, trophic dynamics, and environment, guide the two main questions guiding my work, which is that how does morphological feeding specialization contribute to trophic dynamics and ecosystem function? And how do environmental disturbances influence these trophic relationships? But first, I'd like to put these bigger ideas into an even broader context, which has motivated my research thus far. And that is that there is a fundamental tenet in functional ecology that specialized forms perform specialized ecological structures. And feeding studies in particular have become really classic examples of the tight link we see between morphology and ecology. And so here we have two of these classic examples. Here you see the proboscis of the hawk moth that precisely fits into the columbine nectar spur. And we have the jaw of the egg-eating snake that can expand to at least twice the width of its head in order to engulf this egg. So both of these have become really classic examples of how we understand the tight link between morphology and ecology. But today we're going to talk about how universal this idea really is. Another classic example are the Galapagos finches or Darwin's finches. And I probably don't have to tell this group, but of course, these animals have a wide diversity of beaks that are specialized for crushing small seeds or large seeds, consuming cactus flowers or insects from trees. And what research has shown is that there is a really tight link between feeding morphology and ecology, but there's also this really important environmental component. And so when we think about the small groundfinch, medium, and large groundfinches, this is really seminal work from the late 70s by Peter and Rosemary Grant, late 70s, early 80s, where basically in the late 70s, there was this huge drought that kind of devastated finch populations. And as a result, we see a shift in the distribution of beaks. And so here we have beak depth on the x-axis, number of finches on the y-axis, and you can see before the drought, beaks were shifted around around 9 to 10 millimeters, but that shifts to a larger beak depth because only animals like the large ground finch have the feeding morphology that allows them to crush seeds. And so this shows us that environment really can drive ecological changes. And so mantis shrimp, of course, are another group that are thought to have highly specialized morphology. They've become, they have these raptorial appendages that are used for crushing prey, and they have become very popular in the media, in the popular science media. Here's an example of that. This is a cartoon from the oatmeal. And here's the mantis shrimp smashing the claw off of another crab, and the animal and mantis shrimp saying, and now your dinner. So this is how it consumes its crab prey. And I actually have a video of just this, of a mantis shrimp smashing a leg off of a crab. So here, this is a video from my PhD advisor, Sheila Paddock. And here you'll see the animal smashing the claw off of this crab, and the claw will fly across the tank. This is a high-speed video, slowed down so we can actually see the movement, because you can't see it with the naked eye. So again, this is actually one of the fastest movements ever recorded in the animal kingdom. This movement is producing forces that are thousands of times the body weight of the animal, and that's what allows this animal to smash the claw off of this crab and have it fly across the tank. But of course, this feeding interaction is one of many on a coral reef. And so, but we can look at the separate components of this interaction. And so we can look at the morphology, and then we can look at how the morphology is driving this entire feeding interaction. So if it wasn't for the amazing morphology, you wouldn't have this incredible response from the crab. And so this cartoon is actually really good at showing this, this kind of classic feeding interaction. But as I was saying before, this feeding interaction is small and one of many feeding relationships in nature. So let's scale it down a bit. Mantis shrimp are, of course, consuming many different prey items like you see down here, but they're also consumed by a diversity of predators, and those predators are also consumed by other organisms. And so they're part of this large trophic structure on the coral reef. And then, of course, the environment is critical for governing the diet morphology relationship as well. And in the context of the coral reef, we must consider things like nutrient fluctuations and external, from external oceanographic drivers, seasonal changes in temperature that can alter prey availability, and of course, future changes in ocean conditions due to climate change, such as ocean acidification and ocean warming. Because what the reef looks like today and what these trophic interactions and trophic structure looks like today could be very different from what it looks like in the future. And I think we can use fundamental predator-prey relationships to help understand how ecosystems as a whole will respond to these future ocean conditions. And so again, it's my perspective that we can't understand an ecosystem holistically without deeper knowledge of morphology on the individual level, feeding interactions between organisms, and how this relates to the trophic dynamics and environment on an ecosystem level. So we're going to explore this in mantis shrimp and broaden it to a reef-wide scale. So first we'll look at feeding interactions. So all the kind of amazing diversity of mantis shrimp that we saw on the introductory slide of mantis shrimp is generally broken down into two functional groups. So we have smashers that have hammer-like appendages. They have this bulb at the base of their appendage that they use to smash prey. And then we have spearers with this spear-like appendage that is used to capture prey in between the dactyl and propodal segments of the appendage. Oops. Okay, there we go. Okay, so here we're going to see an animal smash this snail in real time. Okay. So there you see it's smashing a snail. This is real time. And the snail kind of flies across the tank, but of course it doesn't even fully break, which is pretty interesting. So now I'm going to show you another high-speed video of this same movement. And so here we'll see the hammer-like appendage, the dactyl part, rotate forward and smash this snail. And you'll see two flashes of light, one upon impact and one about 11 seconds later. Okay, so here it's going to rotate forward, smash the snail. There's the first flash of light, and the second one is going to show up right about here. Okay, so there it is. So that is called cavitation. So basically, this animal, its appendage is moving so fast that it's creating an area of low pressure that creates an air bubble. When that air bubble implodes, it's releasing energy in the form of light and heat that's comparable to the sun, and that's called cavitation. Because as I told you, this animal is producing movements that are velocities that are comparable to a 22 caliber bullet. The accelerations are yielding forces that are thousands of times the body weight of the animal. And all of this to be able to smash the snail. Okay, and so that was work done by my PhD advisor, but we didn't actually know at the time what the other functional type was doing, and that's the spearing mantis shrimp. And so here we'll see the spear lunge out of its burrow and capture this shrimp on a stick. So there it lunges out, smashes this or captures this shrimp on a stick. And so this again is real time. And so if you look at the linear velocities, you see that they're much slower than the smasher was, but the movement's also a lot different. So let's slow this down so we can see it again in real time, or slow down so you can actually see it, not in real time. So here we'll see the dactyl segments open, and the propotus segment here is going to rotate forward and capture this shrimp between the dactyl and the propotus. And again, so this movement is much slower than what we saw with the smasher, but again, it's a much different behavior. And so when we compare the two side by side, you see that the duration of the spearing strike, again, is much slower than that of the smasher. The speed is about 10 times slower than that of the smasher. And the accelerations are like magnitude slower. But of course, this animal is doing something very different. And when you compare these kinematics to other animals that capture evasive fish or crustacean prey, like these animals do, then you actually see that the movement is quite similar. So these times, these durations are similar to a squid protracting its tentacles or a fish opening its jaws. And so basically this is telling us that the spear just needs to be faster than its prey, but the smasher really needs to be able to produce those high speeds and accelerations in order to yield high forces. So how are these animals moving quickly, both the spear and the smasher? They both have morphology and raptorial appendages that function as a spring. So here's the smashing appendage, and here are line drawings of these appendages. We don't have to worry too much about what everything is called here, but the main thing to note is that the colored elements here, the blue and purple elements, are actual spring elements in the exoskeleton. And so here's the appendage loaded. The lateral extensor muscle has basically pulled the appendage, this smashing part of the appendage close to the animal. There's a little latch here, which you're not seeing, which holds the appendage in place. And when that latch releases, these spring elements literally spring forward, and that makes it so that the appendage flies forward at speeds faster than the eye can see. And so both spears and smashers have this feeding morphology, but they use it in different ways. And so the spear actually has really stiff elastic elements. And so that's why one reason why we think they move more slowly. So that's a little bit about the morphology of the system. Now we're going to think about trophic interactions, and specifically what mantis shrimp eat. And so I told you that smashers are super fast and powerful. They mostly consume hard-shell prey, as far as we know. And they mostly, well, as far as we did know, and they mostly also live in these hard substrate habitats. So they use their smashing appendages for manipulating these hard substrate habitats. Spears, on the other hand, are faster and far-reaching. They consume mostly soft-bodied and evasive prey, and they live in these soft sediment habitats. And so hopefully I've convinced you now that mantis shrimp have highly specialized morphology. for producing high speeds and accelerations and forces. And the question is, does this yield a narrow diet like what we would expect with the classic relationship between form and function, or a wide diet that allows animals to consume other prey items? And so in order to answer this question, we have to know what mantis shrimp eat. And that seems like it's a very simple question. Turns out it's really difficult. You can't just look inside their stomachs and see what they're eating. So I used stable isotope analysis of diet to understand diet breadth over space and time. I specifically studied this animal, Neogonodactylus breedeni. It's a little under an inch long. So it's much smaller than the animals I was showing you before, but you can see that it has a similar behavior. It's able to smash the apex off of this snail. This is my field site in Panama. This is Galeta Marine Laboratory, which is part of the Smithsonian Tropical Research Institute. And it's a really neat field site because there are two habitat types that we see that mantis shrimp live in. So we have coral rubble and seagrass, and we actually find these in replicate plots. And so we can look at how varied diet is over these two habitat types. And so very briefly to begin, we had no idea what mantis shrimp could be eating. And so we did an abundance study where we quantified all of the possible prey that mantis shrimp could be eating. And then with my field assistant at the time, Lovely Avelar, who's now a PhD student in Japan, we did a feeding experiment to look at what mantis shrimp are capable of consuming, given their appendage morphology. And it turns out that they consumed a lot of soft-bodied prey. I'm not going to show you the results of that, but happy to talk about it later if you have questions. But this guided the stable isotope analysis. And so I'll briefly remind this group that, of course, stable isotopes in the predator are a mixture of all the stable isotopes of the prey. And so generally with carbon, we like to say that you are what you eat because you don't see kind of a change between the predator and the prey stable isotope ratios. But with nitrogen, we like to say you are what you eat plus 3 parts per mil, because you see generally 3 part per mil change as you go up the food chain. And we call that the discrimination factor. So that's the difference between the predator and the prey stable isotope values. So to perform the stable isotope analysis, this was my other undergraduate at the time, Julia Hassan, who is dissecting all of the smashers and all of the different prey items. And so we analyze the carbon and nitrogen values. And we also analyze diet with experimentally derived discrimination factors. And so this was my undergraduate, Samantha Liu, who was helping with this work at the time. For those of you who are interested, we found something really surprising, that with mantis shrimp, with this species in particular, we found a 3 part per mil difference, so 3 part per mil discrimination factor between mantis shrimp and their prey, which is not what you would expect. And we found 1 part per mil difference with nitrogen. And so that's actually like the opposite of what you would expect with discrimination factors. And so here, these data are accounting for the discrimination factors. But we have the coral rubble mantis shrimp here in yellow and in this graph, and these are all the little dots you see. And then the seagrass mantis shrimp are in blue. And we have the mean stable isotope values plotted of the prey. And this is the standard deviation. And so the hard-bodied prey are in solid lines, and the soft-bodied prey are dotted lines. And again, this is a bivariate plot of carbon on the x-axis, nitrogen on the y-axis, which is a standard way of how we look at stable isotope data. And the thing to notice here is that consistently in the coral rubble and the seagrass, mantis shrimp generally seem to be falling in this area where they, perhaps we can hypothesize, are consuming more alpheid worm prey, clam prey, and fish prey. But we wanted to know, of course, what are the proportional contributions of each of these diet items statistically. And so we used a mixing model analysis of diet, so based on Bayesian statistics. And the neat thing, don't have to tell this group, but the neat thing about Bayesian statistics is that you can inform your model with prior information. And so we did this. We used 3 different priors, actually. So first we had informative priors. And so based on the literature, we kind of forced this smashing mantis shrimp to be a specialist on hard-shelled prey. And we did that by giving hard-shelled prey 4 times the weight of soft-bodied prey in the analysis. And we also had a prey abundance prior. So we used the abundance data that I had taken previously to build a prior to inform this model. And basically, snails and hermit crabs were the dominant animal. And so that's why you can kind of barely see, but the hard shell distribution is all the way here in the corner, and the soft body distribution is all the way here in the other corner. So we ran the model with both of these priors, but then we also compared it to a prior without any information, what we called a generalist prior. And this is how these models are usually run when you don't have prior information. And so all prey are given equal weights, and that's why we called this a generalist prior. And again, this is kind of the standard way that these models are run. So first I'll show you the no information generalist prior. So we have all of the prey here on the x-axis, and then we have the median proportion of the diet on the y-axis. The first thing you probably note is clams seem to be an important part of the diet. But fish, surprisingly, seem to be a really important part of the diet as well. And so this animal that we originally thought just consumed hard-shelled prey seems to actually include a lot of these evasive soft-bodied prey in their diet. And when we ran the model with our 2 informative priors, the hard-shelled prey specialist prior and the prey abundance prior, we found very similar results. And so in both of these models, you also see that fish seem to be an important part of the diet. Clams and fish, both. But I should also note that the animals are likely consuming the other prey items as well, but there's much stronger support for fish and clams being important parts of the diet. And so this told us that together, this tells us that specialized feeding morphology seems to broaden the diet of a smasher. And this smasher is probably a generalist coral reef predator that consumes a diversity of hard and soft-bodied prey. I also want to note about how I think they are consuming fish and clams and why I think those are important parts of the diet. So it's really important for mantis shrimp to stay in their coral rubble cavities because there's high competition for those cavities. If they leave the cavity, they risk losing it. And so conveniently, a lot of those clams seem to live under or around the same coral rubble species that mantis shrimp live in. So I think it's kind of an easy access prey item for them. And then fish, we think that they are, actually spearing fish, live fish that swim by their burrows, but they're probably also getting some benthic fish and also perhaps scavenging fish that may be fish material that may be around their cavities as well. So now we understand a little bit more about what mantis shrimp are eating. But I also wanted to know, okay, so that's what a smasher is consuming. What would a smasher consume if it was in the presence of a spear? And so we're going to broaden this feeding interaction out just a little bit more. So this is another one of my field sites, the Gump South Pacific Research Station in Moorea, French Polynesia. And if you put on a mask and go for a snorkel, you'll see a lot of habitat that looks like this right in front of the station there. So you see these kind of coral heads, and then you see a lot of coral rubble down here. And in this coral rubble, you'll likely find 2 very small mantis shrimp species, Gonodactylus childi and Ralcerania moorea. These animals are very small, 20 to 25 millimeters. but they have kind of the classic smashing and spearing appendages. And what's really neat, this is one of the few places in the world where we see smashers and spearers living in the same habitat. So both of these animals are occupying coral rubble cavities. And so this allows us to look at diet differences while controlling for habitat. So I did the same stable isotope analysis for these animals. We did not have the luxury of experimentally derived discrimination factors or abundance data for this study. And so instead, we ran the analysis with two different discrimination factors. So one, the mantis shrimp discrimination factor, the experimentally derived ones for breed and I, my original species, and also the literature values. And I'm just going to show you the results from the experimentally derived discrimination factors because the results were quite similar yet again. Okay, so here are the plots. So here we have, this time we have spears in yellow and smashers in blue. And again, here's the median proportion of the diet. Interestingly, clams seem to be an important part of the diet in both groups, but here we see a really interesting shift. And so smashers are consuming a lot of clam prey, whereas spears seem to be consuming a lot of fish prey. And so here we see this classic separation that we would expect, where we have spears consuming hard-shelled prey and soft body prey, and smashers consuming hard-shelled prey. And so this tells us that in the presence of spears, smashers are probably kind of forced to consume these hard-shelled prey items. They may prefer soft-body prey items because they're easier to get, they're more proteinaceous, but in the presence of a spear, they have to use their morphology to smash hard-shelled prey. And so I think we found that both species, again, are consuming a variety of prey, regardless of their appendage morphology, because they are probably including those other prey items in their diet. Morphological specialization really allows these smashers in particular to consume otherwise inaccessible prey. And the diet of these species is likely a result of consistent competition for prey in the past, which is really interesting and an idea that I am very excited to pursue in the future. So that is more about trophic dynamics of the system. But again, I told you that environment is a really important component, and we want to know how environment may shift this diet morphology relationship. And so particularly, I am interested in ocean acidification and ocean warming. We know that ocean acidification affects calcified structures. So here you see classic calcified invertebrates that are affected by ocean acidification or have been shown to be affected in literature. But interestingly, crustaceans have a variable response. And so some crustaceans show kind of, instead of a weaker exoskeleton, more heavily calcified one. Some show a neutral response to ocean acidification. And so we were, for my postdoc, really curious to know how feeding morphology may change in response to decreased pH and increased temperature. So ocean acidification and ocean warming. And really interested to know about this in the context of mantis shrimp, because of course, they are so dependent on their exoskeleton for producing this critical feeding function, which is smashing hard-shelled prey. And so if they are experiencing an exoskeleton that is weaker or even more heavily calcified under OA conditions, that could really affect their ability to really do most of the important things they need to do in their daily lives, like feed and compete with other animals and ward off predators and that kind of thing. So with my two undergraduates at the time, Victoria Morgan and Grace Chan, who are now PhD students at UC Davis and UC Irvine, respectively, we did this ocean acidification experiment where we had mantis shrimp in all of these cups. We had an ambient treatment, a low pH treatment, and a low pH high temperature treatment. So we were looking at the interactive effect of low pH high temperature. And then we had the animals in that experimental setup for 3 and 6 months. We sampled at those 2 time points. And then we looked at exoskeleton structure and mineral composition. And so here we looked at the carapace, which we consider to be kind of like a generalized exoskeleton structure. It's used for protection, but it doesn't have the same importance in terms of being acting as a spring. And so we compared that to the mirror section of the appendage, which is where a lot of the elastic energy storage elements are. And we also, for part of it, looked at the dactyl, which is the smashing surface. But I'm just going to show you the mirrors and the carapace today. So here are the results. We looked at calcium and magnesium. So here, the percent calcium in the exoskeleton of the carapace and the mirrors at 3 months and at 6 months. And we actually saw no significant change and no differences between treatments. And this was similar for magnesium as well, although in the mirrors at 6 months, there was a little bit of an increase in magnesium and the low pH treatment that might be helping to stabilize amorphous calcium carbonate, but that requires further exploration. We also looked at the material properties. So this was my undergraduate at the time, Summer Webb, who's now a PhD student at Scripps. And she is using this nano indenter to basically apply a known force to an exoskeleton structure. And then from that, we can measure the hardness and stiffness of the material. And again, here's the carapace and the mirrors at 6 months and 3 months. And we actually saw no changes in material properties between treatments either. So this tells us that Breed and I seem to be really tolerant to future ocean conditions. What I'm not showing you here is we also looked at stress response. So in particular, the production of reactive oxygen species. And we even tried bringing these animals down to a pH of 7.3, and we saw no change in their stress response as well. So they seem to be highly tolerant to future ocean conditions. And this may mean that they're able to exploit less tolerant species. So perhaps hard-shelled prey that are predicted to be more, to break more easily in future conditions. They could take advantage of them perhaps more readily. And again, if these organisms experience die-offs, then it's possible that even these unaffected prey might be, mantis shrimp might utilize them as a resource more and have an effect on their populations as well. But these again are experiments that I would really like to pursue to know more about how the prey are going to be affected by future ocean conditions and how mantis shrimp responses may influence the trophic interactions on a broader scale. And so all of this work, I think we can summarize with sort of 2 main findings. One is that specialized morphology can broaden diet like we see in mantis shrimp. Also, like, here's a really classic example of that. Cichlid fishes, some of which have jaws that are highly specialized to consume the scales often of another fish. And of course, Galapagos finches are another example of this. And so specialized morphology gives these animals access to inaccessible prey, but it is context dependent because all of these animals will consume other prey that's more available or maybe more nutritious when given the opportunity. And I think that like the new techniques that I used in stable isotope analysis really allowed for learning about the new contributions of diet. And it also makes us wonder if the diets of animals that are considered to be classic specialized feeders, if perhaps they're more unknown. So maybe we just need to look at them in a different way to know how broad or narrow their diets really are. The other thing I think we learned is that mantis shrimp predators may be really important trophic links between macroinvertebrates in the intertidal and larger coral reef predators. So we know now that mantis shrimp consume a diversity of prey and that they are likely consumed, that they have many predators as well in the coral reef habitat. And so I think they may be linking these kind of smaller macroinvertebrates with larger predators. But again, that's work that I really hope to pursue here at San Jose State as well. And so for kind of our main questions, I think the take-home messages are that feeding specializations connect mantis shrimp to a wide diversity of organisms in the food web. And mantis shrimp are probably tolerant to future ocean conditions, which may impact abundances and distributions of prey. So again, I think we were able to learn a lot about trophic interactions and trophic dynamics by looking at morphology on the individual level. And so we learned that morphology and behavior inform broad ecological theory. Trophic dynamics of cryptic fauna, particularly mantis shrimp, might be important drivers of ecosystem function. And shifts in feeding abilities and trophic dynamics may occur with climate change. And so, of course, mantis shrimp are one of many little animals that live in the massive coral reef. And so coral reef, of course, has many little holes in it. And there are a ton of animals, most of which are still unknown to science, living in those cryptic habitats. And so for another part of my postdoc at Scripps, I collaborated with engineers at UC San Diego, and we are in the process of developing a robot that will actually allow us to look inside the coral reef and access those cryptic habitats that are like in the depths of the coral reef matrix. And this is a prototype robot that we made. It needs work, but you know, it's a good start. So we have a GoPro on the end of a sock essentially, and the sock is turning inside out, and that allows it to enter the reef and conform to the reef as it moves through. As a beginning step for this work, I just generally wanted to know who lives inside cryptic habitats and what roles do they play. And so this is work that was done in Maui, where just to get a general idea of who lives in cryptic habitats, I had a GoPro on a selfie stick and also a pH temperature sensor that was extended from a CFET, if you're familiar with those, but a temperature pH data logger. And we were basically sticking those devices into holes in coral reefs. And right now with the undergrads in my lab currently, we are analyzing all of those videos and environmental data to see if we can understand, just get kind of a first glimpse at what's happening inside the reef. And so also really interested in shifts in morphology and trophic dynamics with climate change, as we talked about. This is a very familiar picture to all of you, but this is actually a photo from San Diego at Cabrillo National Monument, for those of you who are familiar with it. And so with my undergraduate at the time, Summer Webb, who I told you about, to kind of start to understand how the role that urchin feeding is playing in the crazy kelp forest urchin dynamics that you all are very familiar with, we looked at how the Aristotle's lantern may change in morphology with feeding abundance. And so there was previous research showing that with feeding abundance, the ratio of the jaw length to the test diameter changes with food abundance. And that's thought to have an adaptive significance, allowing urchins to scrape calcified algae more easily when they are in a starved urchin-barren situation. And we found that actually that they really just eat when they're hungry, which makes a lot of sense. But the question then becomes, how might this change in response to future ocean conditions? So I'm really excited to look at how the Aristotle's lantern may shift with future ocean conditions and how that may influence the kelp forest urchin dynamic as well. And like I told you before, in times of starvation, urchins maintain their Aristotle's lantern, but that's just because they're not laying down new material when there's not enough food. And very excitingly, I'm starting a new project, hopefully soon, As we all know, things are delayed in research in our current environment. But with a collaborator at UC Davis, Brian Gaylord, we recently got a little bit of funding through C Superb to look at how, look at differences in material properties between invasive species and native species, particularly of clams. So Asian clams are very invasive in the bay. And we are curious to know how they cope with large fluctuations in pH. And so, and can these organisms help predict adaptations to future ocean conditions? And so, of course, estuarine animals live in these highly variable environments like we have here in the bay. And so we can look at whether or not that variation corresponds to variation in material properties and start to understand how animals cope with living in a variable environment, and then scale that up to understand how that's influencing trophic dynamics as well. And so to illustrate the variation in pH and temperature, this is just kind of a snapshot from the network of pH and temperature sensors in the bay. This is actually from the summer, but it's to show you the variation that we see in pH and temperature in the bay. And I'm really excited about this next project as well, with Scott Hamilton and Mike Graham at Moss Landing. So very excited to start working down there and meeting you all in person at some point. But we are going to be looking at whether or not the co-culture of abalone and seaweeds ameliorates negative physiological and biomechanical responses to ocean acidification. And so Scott and Mike have found that abalone do grow better. So if you just look at changes in size when they're in a seaweed buffered environment, but we don't know yet how that corresponds to changes in material properties. So are they actually building stronger, more resilient shells? And do we see changes in stress response? So this is an example of the kind of deterioration you can see and dissolution that you can see in abalone shells. This is work that just came out by a group in France, I believe, but it's showing how shell structure changes. So you see more dissolution patterns and in an OA environment, but the question is if buffering will ameliorate that. And so we actually did get funding for this too. So hopefully we'll be starting at some point next year. So again, I think all of this work really demonstrates how looking at morphology on the individual level can scale to trophic dynamics and environment. And yeah, I'm really mostly very excited about getting to work with you all at Moss Landing, hopefully soon. So with that, I will take questions. Thank you. Okay, I guess.
Great, thank you so much. That was excellent. Very, very interesting talk. Gonna take a look at the participants tab here to see if any hands get raised for questions. Maybe, oh, yep, we've got a question from Randy. So I'm going to unmute you, Randy.
Hello. I was interested in, you talked about how mantis shrimp, they're mostly in the reef system. So I was wondering how coral bleaching may play a role in the predation dynamics of the mantis shrimp.
That is a great question. And I think it depends on the species. So I was telling you about 3 species that really like coral rubble. So actually they are okay with coral bleaching because they get like dead coral rubble really, and they're able to build really beautiful coral rubble cavities and kind of manipulate the cavities such that it's this beautiful structure for them to live in. But there are other species that are more reliant on live coral. And there are reductions in their abundance when there is coral reef bleaching, but interestingly, they do seem to be the species that come back one of the quickest when you have recovery from coral bleaching. There's also a really kind of seminal study that was done in the 80s. There was an oil spill in Panama in that same field site that I showed you. And so researchers were able to track the recovery of different species. And again, it showed that mantis shrimp, they come back. They're one of the first to come back. So they're pretty resilient in general.
On the same topic, I kind of have another question. With the high tolerance to ocean acidification, you talked about how they may take advantage in the future of the OA affected prey. And I was wondering, with those prey declining, do you think you would see shifts in the selective pressures on those specialized appendages, like the smasher?
Yes, great question. And I think that that is highly possible, especially if populations, continue to decline for many generations, mantis shrimp generations, of course, then yeah, you might see a weakening of the appendage and the ability to produce these amazing strikes.
So interesting. Thank you.
Yeah, thanks. Good questions.
Thanks. Next up, we have a question from Travis. Should be unmuted now.
Cool. Okay, so Randy kind of stole my question there. But so good question, Randy. But so I guess more generally then, I'm a little interested in, I guess, the competition between the smashers and the spears and how much overlap there is, or if they're even competing with each other at all.
Smashers and spears, yeah. So if you're talking about so mostly they don't live, we don't see much overlap. We don't see much habitat overlap. But in environments like Morea, like you might be thinking of, where they do live in the same habitat, it's highly possible that they are competing, that there is direct competition. And that's actually something I really want to figure out more and study more in the future, because it's highly possible that they're competing for the same coral rubble cavities, because I told you before, coral rubble cavities are really hard to come by. But I think competition for prey is probably indirect. And so I think spears are just much better at capturing fish prey, and that doesn't leave much fish prey and plankton as well, actually, for mantis shrimp, for smashers. And so smashers have to consume the hard-shelled prey. Thanks. Good one.
Interesting question. Next up, we have Alex with a question. Unmuted.
Hi, this is a really wonderful talk. Thank you for giving this seminar. I thought your finding that if the smashers and the spears were not co-occurring, they were both effectively generalists, was really interesting. But I'm having a little trouble understanding how something with a spear morphology can eat hard-bodied prey and how something with a smasher morphology can eat soft-bodied prey.
That is a really good point. And something actually that I usually show you when I'm in person or have the video on, so apologies for that. But the spear does have, the smasher does have generally a really sharp tip dactyl. They don't have the same morphologies, they don't have the same spines here often that smasher spears do. But I've seen them stab prey before, and they're definitely not as, even though they're incredibly fast, they're just not as elegant at it as a spear. But they also swim up to hard, soft-bodied prey that doesn't often swim as fast, and they smash it as well. So I've seen them like take on a worm that way, and even like a alphiid snapping shrimp. So they'll like swim up to it and like smash, 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 and then subdue the prey that way. Spears, on the other hand, they generally, we think it's only probably the larger ones. And also, although the smaller guy I showed you did have interesting spearing appendage, it did have kind of your typical spear, but it also had a wider dactyl, and we think that's probably helping it to smash. But then the larger spears, they can bring hard-shelled prey into their cavities, and we think that they are able to smash clams, maybe not like super heavily armored snails, but they are able to access it. It just takes them a lot longer, and they do it in their burrows so that they can work on it for a while. But
Cool.
yeah, they're not big parts of the diet.
Okay, cool. Thank you.
I was wondering the same thing. Thanks for asking that. Like, how does it work when you don't have the strong smashers? So thanks for asking that. And then I got a message. We have a question from Jacob as well. So I'm going to unmute Jacob now. There we go. You should be able to see.
Okay. Thank you for your presentation. I really liked how you showed differences in prey selection with the combined combined styles, and then how the environment would change that prey availability. How do you think prey selection or just preference would change with the density of the shrimps themselves? Like, if the environment affects the abundance or their habitat availability itself, how might they partition different prey selection that way?
Interesting. Well, yeah, so I can tell you that work from the 80s, again, showed that mantis shrimp have a significant effect on snails and hermit crab abundances, actually. And so where they were able to remove mantis shrimp from the environment, and also compared mantis shrimp removal plots to mantis shrimp present plots, and they found that mantis shrimp were affecting the snail and hermit crab populations, which is actually pretty interesting, since we now know that that's not like a main part of their diet. So we know that but they are affecting hard-shelled prey. And so the question you asked, though, what does that mean for the future? Yeah, I mean, I think that's really what I am interested to study more. And so I would love to be able to put mantis shrimp and their prey items in acidified conditions and look to see how much more easily they consume these hard-shelled prey items, and then kind of extrapolate those lab experiments to what could potentially happen in the field in the future. I don't know if that's a very good answer to your question, but I feel like the jury's still out.
Great, thank you. Oh, so many great questions. Thanks, guys. We have one more hand raised. I think we should take that question, and then I'd like to ask one before we break into more informal discussions for happy hour, if you're okay with going over time a little.
I mean, I am.
Cool.
So
Okay, great. Okay, so next up is Jackson. And oops, I almost got you unmuted, and then it. Maybe I need Alora's help with this one. Sometimes it just doesn't click for me, Alora. Okay, you're good now. Go ahead, Jackson.
Hello. My question was, so I saw that you had as predators of this mantis shrimp, like octopi and sharks, and I was wondering how they play into these changing conditions, if you knew if they were gonna exert more predation pressure on mantis shrimp or less, or is that not gonna change much? And like the mantis shrimp, they're kind of the winners in climate change as well.
Yeah, definitely. I think mantis shrimp are definitely going to be one of the winners, quote unquote, with climate change. That is a really great question. And I also future direction to look more at the predator side of things. I haven't really done that. And like what changes or lack thereof in morphology and mechanics might mean for defense strategies. But I could see, if over evolutionary time, a mantis shrimp appendage is becoming weaker because they don't have to smash hard-shelled prey in the same way, probably their defenses may go down. However, selective pressure from predators and selective pressure from needing to manipulate their habitat, but also compete with one another for space, for cavities, could also mean that the amazing capacity of their appendage doesn't change. And so even though they might not have the same selective pressure from needing to eat hard-shelled prey, presumably they might still be able to produce really amazing smashing strikes because they're still competing with, there's still interest-specific competition in the field, I guess. But yeah, the question about predators could still be an important selective pressure. And I think that's a great future direction too.
Thanks.
Cool, great. Looks like we did have one more hand raised actually from John Geller. So I'm gonna unmute John. Okay, it looks like it's good now, John.
Yeah, hi. Very nice talk and nice to meet you. My question, I've always, found it fascinating the specializations in the skeleton of stomatopods and snapping shrimp that are necessary to generate such high forces and speeds of the appendages. And I was wondering, in this case, what the spring-like materials in the claw or the dactyl of the stomatopod is made of.
Okay, yeah, great. So we actually know a lot more about the dactyl, so the smashing surface, than we do about the Mirus segments, so the elastic energy storage units. As far, from what I know from my work and from work from my PhD advisor and others, the material in the Mirus and the elastic energy storage elements is kind of like different proportion of calcium to magnesium and chitin with a bit of resin as well. The dactyl is really neat. There's an amazing paper that came out, I can't remember how many years ago now, but it's showing that it is a multi-layered, multi-structured appendage. And so there's like different layers of material piled on. They have the classic, helicoid structure that you see in exoskeleton, but then that's coupled with like increased chitin and increased calcium carbonate actually. So it's this really like fortified structure.
So those, in your diagram, you had two springs highlighted. So those are part of the exoskeleton?
Yes.
Okay.
Yeah. So there's the mural V and the ventral bar, which are both exoskeleton, parts of the exoskeleton,
like, infoldings from the inner surface of the skeleton, or those?
Yeah, let's see if I can find a good.
And
yes.
is there anything like that in snapping shrimp, or is that uniquely stomatopod?
So snapping shrimp do also have a spring-loaded mechanism. And so they have kind of elastic energy storage elements, but they have a latch system that stays open as far as I know. The mantis shrimp don't do that. It's muscle controlled. So they kind of engage their latch just like it's like a couple milliseconds before they smash. So right, when the snapping shrimp are doing it, it's a latch mechanism that's spring-loaded that they then can shoot their cavitation bubbles with. But mantis shrimp, they're producing cavitation because they're just moving so quickly through water. So yeah, I think, and I actually don't remember off the top of my head this comparison of speeds between the snapping shrimp and the mantis shrimp. Mantis shrimp are faster, but I don't know by how much, because the snapping shrimp creates an area of low pressure by like clapping their claws together. But with this mantis shrimp, it just is the full movement. I don't know if that doesn't really answer your question about materials, but it maybe perhaps answers the comparison between smashers and snapping shrimp.
I was just wondering, like, more broadly in the crustacea, we don't see that sort of behavior very widespread. It's a very, as you premised your talk, it's a very specialized morphology. And so I was just trying to think, what are like the evolutionary limitations that have to be overcome for that trait to evolve, because it is so unique.
Yeah.
So material might be one aspect of it.
Definitely. And then also, in evolutionary history, we start to see smashers kind of the smashing appendage evolve and spearing and smashing kind of splitting around the time when we start to see coral reefs forming and in large abundances as well. So that happens, yeah, around the Triassic or so. So there's definitely a habitat component to the evolution of the strike, I think.
Thank you.
Thanks.
Cool. That's great. I would love to continue. I'll save my question for the social period, just for those who need to head off after this. But thank you so much for such a really interesting conversation starting seminar. And thank you everyone for being here. And please feel free to stick around. Turn on your cameras and microphones if you want to stick around and chat about all things stomatopods and smashes and spearing things.
Yeah, thank you guys so much. It was really, really fun to get to talk to you all. Great questions.
I wanted to jump in just about snapping shrimp a little bit.